Good morning.
Good morning.
Morning, everybody. Happy Friday.
Morning.
Nice little snowy Friday for us. All right, we're right at 11:20. Go ahead and type your name, your first name into the chat for me so I can take attendance if you haven't done that already. Good to see everybody here. Today, we're going to be finishing up our discussion of gram staining, and specifically, we're going to be practicing looking at some slides, some smears that have been gram stained, and we'll practice interpreting those. Before we get started in that, though, I want to talk just a minute about a story that's all over the news right now, which is what's going on down in Texas. It does have a little bit of application to microbiology. As you probably have heard, they're having a winter storm event in Texas. They're having an unusual period of cold weather, and their power grid has essentially collapsed. People have been without power for several days, although it is coming back now. And predictably, after not having power for a couple of days, like statewide, the water treatment plants have gone offline. So a lot of Texas right now is under what's called a boil water order. I can't imagine how much those folks are suffering. We all know here in New Hampshire how miserable it is to be without power in a winter storm. It's just miserable. And then on top of that, to have your water supply cut off, very, very difficult. And of course, they're dealing with broken pipes because nobody's homes are winterized down there, and on and on. But anyway, the application of this story to microbiology has to do with that boil water order. Does anybody know, or could you make a guess, how long you would have to boil an unsafe quantity of water, something that has not been properly treated, in order to make it safe for human consumption? If you know, go ahead, type it in the chat for me, or if you want to take a guess. How long do you think you'd have to boil a pot of water to make it safe to drink?
2 to 5 minutes.
Good guess. Sean says 3 to 5. That's a good guess. Anybody else? Do you have any sense of that? Most people don't have a sense of how long to boil it. Ruth says 10 minutes. That's also a good guess. We have to remember what could be in our water if it has not been properly treated. We're talking about bacteria, right? We're also talking about fungal organisms, perhaps algae, perhaps protozoa, and bacterial spores and things, right? Brianna guessed 15 minutes, and Brianna wins the prize today. You have to actually boil water for 15 minutes to make it safe to drink. And unfortunately, most people don't know that. I mean, why would you know that if you're not accustomed to boiling your drinking water? And one of the areas where public health and the news media always make a mistake is right here. The public health authorities will make the announcement that the city or the town is under a boil water order, that everybody needs to boil their water to be safe. The media picks up on that and reports that, but nobody talks about the length of time. So there's a fair number of people who hear that and think, oh, I have to bring the water to a boil and then I can drink it. And of course, that's not nearly long enough. Yeah. So this comes up all the time during emergencies that municipalities, because of a power outage, have to shut down water treatment facilities. And as a result, they can't say for sure that the water in the system is safe. A lot of what happens at a water treatment facility is just there's no magic really. It's water that's being allowed to have materials settle out of it. But as it passes from place to place along the line in the water treatment facility, that's when chemicals get added and that's when filtration takes place and so on. But once the electrical pumps stop working, now nobody knows if it's safe. What's in the line may not be safe. The other thing to remember is when water sits in lines, in water delivery lines, it becomes dangerous from a microbiology standpoint as well, which is why if your water's been shut off for a certain amount of time, they also suggest that you boil your water. And then when the water service comes back, that you flush your pipes for a few minutes so that you know you have nice, fresh, treated drinking water. But yeah, spare a thought for those poor people down in Texas, because while electricity is returning now, which is wonderful, now everybody's pipes are breaking. Just a mess. And unfortunately, something that was preventable in the long run. So yeah, a little bit of information there from the news about microbiology. All right. The other thing I wanted to mention was that your first lecture exam has been graded, and you may or may not have seen that grade posted, but those are up for you to see. I was very pleased with this first exam. The average came in at a 71 out of 80. Remember, this exam was out of 80 points total. And 71 out of 80, that's a good grade. That's an 89. So that's a very good grade for a first exam. I was very pleased. If you did not do as well as you would have liked on this first exam, don't despair. You have several other exams that you'll be taking, both lecture exams and lab practical exams this semester. So if you didn't do as well as you wanted to on this first exam, don't worry, you'll have plenty of opportunities to make that up. But take this experience as a sign that you should change up how you're working. You need to change up how you're watching the videos, how you're taking your notes, how you're reviewing your notes, and so on, so that you're prepared when you sit down to take the lecture exam. Again, it's not unusual for some students to do poorly on the very first exam. You're not used to me. You don't know the depth of the questions I'm going to be asking and so on. So again, if you didn't do as well as you would have liked, now is a good time to change how you're approaching the material so that you can do better next time. But overall, I was really pleased. We had a couple of students earn a perfect grade, which is great. And we had a lot of students in the A and B range. So very, very good. Good work. All right. Looking ahead to next week. In fact, I'm going to go ahead and pull up a screen to share with you. This is just our schedule. Remember, this is at the end of the syllabus. If you ever want to see our schedule all laid out for the semester. So we're right here. We're going to be finishing up the gram staining exercise today. Looking ahead to next week, you have your first lab practical exam on Monday. I'm going to pull up the instructions in just a moment that you're going to see when you take this exam, because I want to show you something regarding how to take it. The lab practical exam is going to feel a lot like the lecture exam in that you'll need to take it sometime between 8 o'clock in the morning and 8 o'clock that night. So you'll need to find a block of time where you can sit down and have 90 minutes time to complete the exam. Again, most students don't need all 90 minutes, but you'll have that time available if you need it. And of course, if something comes up over the weekend or something, and you realize that you are unable to take the practical on Monday, please send me a message and let me know, and we will figure out a makeup time for you. Something else to see right here on the schedule, there is no lab meeting on Monday. So lab practical exam on Monday, but we will not be getting together for a lab exercise on Monday. So make sure you've got that jotted down on your calendar. We will meet on Wednesday and Friday next week. And we'll have a little bit of a break in that we're not going to be doing an exercise that will have homework associated with it. But instead, we're going to be discussing the SARS-CoV-2 virus. And of course, this is the virus that is causing the current pandemic. And it is the cause of COVID-19, the disease called COVID-19. You're going to be viewing two different lecture topics next week. One of them is eukaryotes, and the other is the viruses, or more precisely, the acellular microbes, the microbes that are not cells, but rather particles. And that includes the viruses. So it's a nice opportunity for us to just talk a little bit about this scourge of SARS-CoV-2, which has been impacting all of our lives over the past year. There won't be a homework assignment associated with this discussion, but you may see some of what we talk about show up on a future exam. So you'll want to be sure to attend that lecture, or that discussion, I should say. So that's coming up next week. Now, the next thing I want to share with you is our Blackboard page. So I'm just pulling up our page, our course content page on Blackboard. That's what you should see in front of you right now. I'm going to scroll down and click on next week, which is the week of February 22nd. Here's our to-do list right at the top. And then you can see a lecture quiz for each one of the two lecture topics, the slide sets as usual. This is what I really wanted you to look at though right here, and we'll just talk about this for a minute. The lab practical exams are just exams on lab material. So it's going to look very similar to the lecture exam in terms of, how the questions will be asked and so on. It's just that it's not based on lecture material. It's based on the things that we've discussed in our Zoom labs. You are allowed to use the notes that you took during our Zoom labs, that you're taking during our Zoom labs. You're allowed to do that, just like you were allowed to use your lecture notes during the lecture exam. But of course, you're on your honor not to use any other materials during that exam, including getting help from someone. Now, I've added this line in here. If you use prohibited materials to answer a question, you will receive a 0 for that question. I just want to remind everybody that there is an academic honesty policy here, and we all follow that. I know that it's very, very tempting to Google questions while you're taking an exam remotely. I know it is. It's very tempting when you don't know the answer to just type it into a search engine and find the answer. But that's cheating in this course. You're not allowed to do that. All right. So please don't risk getting caught doing that. All right. There are ways that professors like me can tell if you have used a source other than your notes during the exam. So please don't risk that. It is so much better. If you don't know the answer to something, it is so much better to get it wrong and lose a couple points than to risk getting in trouble in terms of academic dishonesty. You don't want that to happen. Trust me, you don't want to get referred up the chain of discipline for academic dishonesty. Now, here's where the lab practical exam looks a little different from the lecture exam. The questions for the lab practical exam are actually going to be written in a file. So when you come here on Monday, you're going to click right here on where it says questions, lab practical exam 1, and you're going to see the list of questions. There are 12 questions on the lab practical. Now, you're going to type your answers into the quiz, just like you always would. You're going to type your answers right into the quiz in Blackboard, and I'll show you what it looks like in just a minute. But the questions themselves are written on a separate document, and here's why. Lab practicals often contain pictures, right? Pictures of things that you're going to be asked questions about. And if you think about what we've been talking about in lab, gosh, it started with things like biosafety and the microscope, and we've looked at slides with organisms on them and things like that. We're looking at gram-stained slides today. You're going to be shown images and asked questions in some of these questions. So that means those images have to be in a separate document. So we just put the questions and the associated images into its own file, which you open up in a separate window, and then you're going to type your answers right into the quiz like normal. Just a couple things to note, just like with the lecture exam, you have 90 minutes. You can certainly go beyond 90 minutes, but you might lose a few points of credit. Most students finish this exam in about 30 to 60 minutes, so you should have plenty of time. It'll open up at 8 o'clock. It's due by 8 p.m. Okay, again, you can submit it past 8 p.m., but you need to get in touch with me if you need to go past 8 p.m. And once you start the exam, you have to finish it. You have one attempt at the exam. And when you finish it, remember, you've got to click at the bottom where it says submit the exam. Don't forget that. A couple of you have learned the hard way that when you finish a test or a quiz or anything else on Blackboard and you don't click that submit button, number one, I don't receive your work. And number two, your work gets lost. So if you go in and try to submit it, you'll have to retake the exam again. So don't forget to push that button at the end. And as always, if you have any problems during the exam, any technical problems, just shoot me a message and we'll figure it out. All right. No need to panic if something happens. Just shoot me a message and we will figure it out together. Now, I want to show you what it's going to look like, again, when you come in on Monday. Remember, you're going to click here to get the questions. And then when you scroll down, you're going to see the places where you're going to type your answers. So you can see here for question 1, it says answer question 1 here. And there's A, B, C, D, E, F, G, H on question 1, okay? So you're going to be typing your answers in here, but you're going to be reading the questions in this document here, okay? It's just a little bit different from the lecture exam, again, because we use more images in the lab practical portion of the course. Okay, any questions? Any questions I can answer? You're free to have your notes with you, the notes that you've taken during lab. So those can be right by your side. Hopefully that takes a little stress away for you. All right. Okay. All right, if there's no questions, what I'm going to do is now pull up where we left off when we were talking about gram staining. And like I said, we're going to look at some slides and we're going to get some practice reading these gram stain slides. Remember, when we make a smear and we gram stain it, the reason we're doing that is because we want to figure out what the gram status of this organism is. And we said gram status is defined as, or, reported as, whether it's gram positive or gram negative on the slide, purple or pink, and what shape the cell is. So maybe I might have a gram negative coccus, or I might have a gram positive rod on the slide. That's how I would report the gram status. All right, very good. So last time we were together, we finished up somewhere around here. This is one of the slides we looked at that lists the steps of the gram staining procedure, remember, top to bottom for you, so you have those written down for your notes. And then we actually walked through a demonstration, a series of images showing how we go from start to finish performing a gram stain, and which chemicals get applied and how long, and how we create this slide that has had this differential staining procedure done, known as gram staining. When we finished up last time, we looked at this slide. And again, this was a mixed sample here. Remember, this is under the 100x lens. So we're at 1000x magnification, which is the highest magnification we have available to us on our light microscope. And this is what we want to use when we're examining smears. And again, you told me last time we were together that you could see both purple cells here and you could see pink cells. So we have two kinds of cells on this smear. We have two organisms. One of them is this circular or coccus-shaped organism, and that one's gram positive because it's purple. And then we also have this rod-shaped organism, which is gram negative because it's pink. And then the last slide we looked at last time was this one. Again, a mixed smear. Two organisms are visible on here, a gram positive rod and a gram negative rod. So, this could be a patient sample. This might be a pure culture that got contaminated, and that's why there are two organisms in it. It could have come from anywhere. Maybe it's an environmental sample, so it has two organisms in it. But remember, when we report what we see, we want to say we see two organisms on here. One of them is a gram positive rod, and one of them is a gram negative rod. Oh, and of course, you could use the word bacillus instead of the word rod. That's fine. Those two terms mean the same thing. So let's take a look at this slide. You've got two smears here, images from two smears. The top one and the bottom one are taken at different magnifications. I put this in to drive home this idea that this image right here was taken under the 40x lens. So we're at 400 times total magnification. This is a purple circular cell. You can just see that if you look closely. Let me enlarge it for you here. Obviously, you can get more detail if you go up to the 100x lens, but you can see what you have at 40x. This is a coccus shape. This is a circular round shape. They are dark purple. This is a gram positive coccus. And if you remember, I told you last time that the other information we can get while we're gram staining is sometimes we can see how these cells arrange themselves in the culture and on the smear. This happens to be a Staphylococcus species, what you're looking at here. You wouldn't know that, of course, but this happens to be a Staphylococcus. And this isn't the best example of this. I have other slides that are better, but Staphylococcus tends to cluster. So if you use your imagination a little bit, you can see that there are these almost circular clusters of many cells over the surface of this slide. Here's a nice one right here. That's a cluster of cells. People call these grape-like clusters. See that one right there? It just looks like the cells are kind of hanging out together. They are arranging themselves. In other words, I don't see a whole lot of individual circular cells on this slide. I don't see them all spaced out individually. They tend to be grouped in these clusters of cells. This is just a feature. This is a characteristic of Staphylococcus, just something that it tends to do. And if we know this feature, this tendency, it can help us when we're trying to figure out what organism this is. We suspect, in other words, that it might be a Staphylococcus. Now, I want to point something else out to you here. Notice how I've written out this name. It says Staphylococcus, and then it says SPP. Remember, Staphylococcus is the genus name. We always give the genus name a capital letter. Notice that there's no species name here, though. Instead, I have this abbreviated SPP. Anytime you see that, this is just a shorthand abbreviation for the word species. So if I'm talking about an organism, but I'm not going to identify specifically what species it is, I write SPP period. Staphylococcus species. It's some species of Staphylococcus, but I'm not going to name exactly which one it is. Down here, you could see it's listed as a Bacillus species, but I'm not telling you exactly what the species is. This image is taken under the 100x lens, and that's 1,000 times total magnification. And I think you can see pretty readily that these are rod-shaped organisms. I told you to try to find an individual cell so that you could tell what shape it is. And you can see one rod right there and one right there. Here's two rods that are head to tail. Here's another two rods, head to tail. If you look across, there are some chains of rods. That can be a characteristic feature for an organism, arranging itself into chains. It doesn't happen to be for this organism. It's nothing that we would expect to see necessarily with this organism, but sometimes the chains, when you see them, are very obvious. And it turns out to be a feature, again, of that particular species or genus. So these are gram-negative rods because they're stained that bright pinkish red color. All right. So what do you see? What do you see on this slide? If you were looking through the microscope and we're under the 100x lens here, and your job was to write down the gram status of this organism, what would you write down? You can
Gram-negative coccus-shaped organisms.
Okay, not gram-negative.
Oh, positive, my bad.
That's right, positive. Dark purple, bluish purple, gram-positive, and coccus-shaped. Good. Do you think these cells are relatively individual across the smear, or do you see any grouping? This is not part of gram status. I'm just asking you as a student of microbiology.
I see grouping.
What kind of grouping do you think you see? Would you say clusters or chains?
Um, Sometimes they look like clusters, like there's an area, especially in the middle, where it looks like clusters, but
Yeah.
then there are areas that look like chains, so I'm not sure.
So this would be a nice example of clustering. Obviously, it doesn't look the same all over the smear, does it? It doesn't look like nice, even, very obvious clustering, but we definitely have clustering going on in this smear. Again, that's not part of gram status or anything, but that's something that we can make a note of if it's our job to try to figure out what kind of organism this is. It would make us think, at least as a possibility, that this might be a Staphylococcus, because we know Staphylococcus is a gram-positive coccus. Now, there are lots of other gram-positive cocci, so we would have to do further testing to be sure. But at least we could start to think in the back of our minds, this might be a Staphylococcus, because I think I might see some clustering here. Good. All right, let's take a look at another one here. This one is also from a 100x lens. We're 1000x total magnification. I will tell you, this gram stain smear was made from a patient sample that was body fluid, not culture media. So the background of this slide has picked up some staining. That's because it has probably some protein in it or something. This is some kind of a body fluid that these microbes are in. So you have to figure out, is it gram positive or gram negative, and what is its shape? So color is first. And then you want to associate that color with either gram positive or gram negative. And then think about the shape. What do you think? What's gram status for that? Let me pull it up again. What do you think? Say it again.
Gram positive,
Yep.
coccus shaped,
Yep.
and chain grouping.
Yeah, there's some real chaining here, right? Does everybody see that? These cells don't like to be by themselves. They want to be with other cells in culture. Now, some of the chains are very, very short. Some of them only have 2 cells in them, but some of them are really long and very obvious. Yeah, big, long chain of cells. They are arranging themselves head to tail. And they're basically, this is just, again, a characteristic of this organism as it grows. You have to remember, these are living things. And just like all living things, they live in communities and they communicate with each other, and they're in a population. And so they behave in certain ways as a population. And one of the things this organism does is it forms these chains. This is a streptococcus. So this is a classical chain formation. This is the kind of chains we look for if we want to call something as arranged in a chain. But remember, the gram status would be gram positive coccus. Good. 2 slides on this one, or I should say 2 smears. On this one, let me get this out of the way for a sec. Now, we are only at 40x here. Hopefully you're starting to realize that at 100x, it's a lot easier to see than at 40x. But I think you can still see some detail in here. I don't know if you'll be able to call shape, but what would you call for gram positive or gram negative?
Positive.
Positive, dark purple, gram positive. The reason I put this slide in here, these happen to be caucus shaped, although like I said, you really can't see them clear enough here. You'd have to go up to 100 to see that. But this is what I would consider a very classic example of that clumping or clustering behavior that we see in some organisms. See how the cells have grouped themselves together in these clusters. And again, a lot of people call these grape-like clusters. They look like a cluster of grapes that you would find in the supermarket. This slide has a nice example of that. So again, I would not expect you to see that these are caucus shaped because we're not at a high enough magnification. But this is definitely the color of a gram-positive result. And then we have this one over here. This one was taken from a patient. Look at the difference in the background between this one that we looked at a minute ago and this one over here. The background over here is just white. It's transparent. There's no color in it at all. This is what we expect to see when we take cells out of a culture and put them on a slide and make a smear. But when we take them out of, say, a sample of tracheal fluid or an abdominocentesis tap, or a urine sample, or perhaps even blood sample, you're not going to have a perfectly clean background because that body fluid has molecules in it. So they're harder to read sometimes when they come in the form of body fluid samples. But having said that, I think you can see a cell here. I think you can see the shape and the color. What would you call gram status?
Positive.
Positive.
Gram positive, and remember, cell shape too.
Caucus.
Caucus. Good. We've seen a lot of gram positive cocci today. Let's switch gears a little bit. Here's a nice example. We are under the 40x lens here. This is an organism that is just enormous. This is a huge organism. Normally at 40x, again, remember this one's at 40x. We don't expect to see shape nearly as nicely as you can here, but this organism just happens to be very large. So take a look at this one. Take a look at the color, take a look at the shape of the cells. And again, just for extra information, take a look at the arrangement, or if you see any arrangement. We don't always. What do you think about that one?
I say it's gram positive.
Gram positive. And the shape?
Chain.
Remember, we want the shape of the cell.
Bacillus.
Bacillus or rod, right? Bacillus or rod shape. So right here, there are two rods side by side. I can just make out that there's two cells here, and they are I shouldn't say side by side, they're head to tail. Okay. Here's one cell right there. Here's one cell right there. One cell. Here's another one. So these are gram positive rods, or you could say bacilli or bacillus. They are in chain formation though, aren't they? They are definitely associating with each other. They're not just randomly spread over the surface of this smear. They are hanging out in a particular arrangement, and it happens to be a chain. This is classic for this particular organism. And I have the name down here. This is Bacillus megatherium. Mega meaning large or huge. This is a very big rod compared to some other rods that we see. So again, gram status would be gram positive rod or gram positive bacillus. Remember, those two words mean the same thing. this smear right here that I'm playing with, this is taken at 40x. Again, it's a little hard to see the cell shape, although I think you can here. But if I was working with this smear and I got under my 40x lens and I peeked in, I could certainly tell the color, even if I couldn't see the shape quite yet. But as I'm enlarging this image, I think you can see the shape here. So somebody who hasn't tried yet, what do you think? What would you report that gram status as?
Gram negative.
Gram negative. And don't forget the shape.
It's rod shape.
A rod.
Rod.
shape.
It's a
Yeah.
rod. These rods are a little stubby, aren't they? They're not nearly as nice and elongated as these are. These rods are a little stubby. This is E. coli. The genus name is written out for you down here. It's Escherichia, but we all refer to it as E. coli. This is a characteristic shape for this rod. It's very stubby. It's hard to, you don't automatically get the sense that, oh yeah, that's a rod. You have to look a little more carefully to see that it's a rod. Do you see any arrangement here? Just out of curiosity? Do you see clusters or chains?
Clusters.
Maybe a cluster right there, yeah. Maybe one right there. But across a lot of this smear, I don't really see too much clustering. It turns out that E. coli doesn't have an arrangement that we see again and again. It doesn't have a classic arrangement that we expect to see. It's just a feature of that organism. Let me get that one down so we can take a look at this one. This one is taken under the 100x lens. What do you think? Somebody who hasn't tried. What do you think? Be brave, speak. Figure out the color, figure out the shape. Gram positive, remember, is purple. Gram negative is pink.
And rod shape.
Yep. What do you
And
think about
still some slight clustering.
Yeah, there's a little bit of clustering here, isn't there?
It's
Gram-
gram negative.
Say again?
Is it gram negative?
It is gram negative. Now you might say to yourself, gosh, how come this pink color is so different from this pink color? Over here. Oops, I don't need that. This pink color is so much lighter, and it's almost a rosy pink or a paler pink. And this one is more like a fuchsia color. How come? The answer is they're two different organisms. And they just pick up these stains a little bit differently sometimes. Your job is to figure out, is it a bluish purple like this one? Or is it a pinkish red? So you're going to see a range in that color from time to time. But you need to, you learn how to differentiate something that runs from maybe pink to red versus something that runs from maybe blue to purple. Okay. They all look just a little bit different when they're gram stained because they're different organisms. Now, I want to drive home the idea that it's not super easy to see things under a 40x lens, but that doesn't mean we can't see them. So we just saw this one. This is that E. coli slide. We said this organism is a gram negative rod. And this is the Bacillus megaterium, gram positive rod. This one, you know, you can certainly see the color at 40x, But if you were looking at this, you might want to go up to the 100x lens to be sure you have a rod. Over here, you can definitely see the color at 40x, and I think you could definitely see the shape at 40x. It just depends on the organism. That's why we say that we need to be at either 40x or 100x to see bacteria and to determine gram status as well. Because for some organisms, 40 is enough. And for other organisms, we need to go up to 100. All right, I'm going to switch gears here for just a minute, and then I'm going to show you a couple more slides. I want to talk for just a minute about the limitations of this technique that we call gram staining. The number one limitation of gram staining is that it's best done with a fresh bacterial culture. Now, what do I mean by fresh? I mean less than or equal to 24 hours old. So if I have microbes growing in the incubator and I want to gram stain them, I need to get that gram staining done while those cells are young. Remember, we normally grow cells in the incubator routinely for about 24 hours. So it's perfectly fine for me to take some cells after 24 hours in the incubator and make a smear and gram stain it. I'm going to get good results. But if I have an old culture, and we typically say cultures are getting old once they're about 48 hours old. If I have an old culture, what's going to happen is I'm going to get some inconsistent staining results. And I'm going to show you what this looks like. I'm just not going to see a very clear difference, a very clear either purple or pink. I'm just not going to get that. I'm going to see maybe some cells are purple and some look a little pink, and some of them look halfway between purple and pink. Once the culture starts to get old, and again, we define that as being maybe 48 hours or older. Once that container full of media and growing cells gets to be old, think about what's happening. That container has a limited amount of space in it, right? And it has a limited amount of nutrients in it for these organisms. And they are dividing. They're growing and dividing. So what happens is they start to use up all the nutrients. And remember too, they're secreting waste materials into that container too. So the organisms are getting old, and they're under some very stressful circumstances. There's not enough nutrients anymore for everybody. There's too much waste around them, and they're starting to suffer. Some of the cells will actually start to die. If you have a bacterium in there that's able to sporulate, it will start to sporulate because it's stressed at this point. Remember, we said the genus Bacillus and the genus Clostridium are two types of bacteria. Specifically, they can be pathogens that will tend to sporulate as cultures get older. Those are the only two genuses of pathogenic bacteria that are capable of sporulating. And as a culture gets old, if one of these two are growing in there, they're going to start to sporulate. That's going to interfere with my gram staining. Remember, gram staining stains the cell wall. It stains the peptidoglycan very specifically. And as a cell begins to sporulate, the cell wall is going to go away. So spores don't gram stain. They just don't. So I'm going to look at that slide and I'm going to just see a very confusing result. Now, if I pulled some cells out of culture and I tried to gram stain them, and I got a very confusing looking slide and I didn't know how to interpret it, I would say that I was unable to determine the gram status. That's how I would report that. And what I would do is I would take some of the cells out of that old culture and I would transfer them into a nice fresh tube of media, a nice fresh batch, so they could start growing again and I could maybe get another gram stain tomorrow. So that's the biggest limitation. It works best with fresh cultures. Now, here's another thing to keep in mind though. All of the gram stains, the crystal violet, the Graham's iodine, the decolorizer, and the safranin, they have expiration dates. All stains, it turns out, all stains in the microbiology lab have expiration dates. And so old stain, expired stain, is not going to perform well during gram staining. And you're going to get bad results with that too. So it's really important every time we gram stain and we pull those bottles out that we check that expiration date and make sure we're using stain that is in date, because it won't work very well for us if it's out of date. Luckily, as I told you, these stains are not very expensive. So we just throw away the ones that expire and get some fresh stain. And remember, we always discard them as hazardous waste. We don't put them down the drain. You know, busy labs, laboratories that have a huge volume of samples coming through and somebody's gram staining all day, every day. Those busy labs go through a lot of stain. The stain doesn't have a chance to expire. But some labs, you know, they're not doing that much gram staining and they buy those liter bottles and it takes them a while to get through them. And before you know it, the stain is expired. And somebody has to be keeping track of that in the lab so that you don't accidentally use an expired stain. It just won't give you a good result. So if you look at a smear, a gram stained smear, and it's not clearly blue and not clearly pink, you can't determine the gram status. You just have to say, can't be determined. I can't see a clear result here, so I can't determine the gram status. Let's try again. All right. Take a look at this one. I think you can see, if you compare this slide to, say, this slide, the color on this slide is not very dark. You definitely get a blue sort of feeling from this slide, or a light purple kind of feeling from these cells. But it's not that nice, obvious, dark blue purple. It's a little bit lighter purple. And if you look really carefully at this slide, like here or here, some of these cells are pink. Some of them have stained pink, and some of them are that light colored purple. This is an old culture. This is a smear that was made with an old culture. The other thing I want you to notice is this. There's something right there that doesn't look like a cell. There's a big glob of something right there. It's black. I don't see a nice, clean, white background on this. This is a culture sample. It wasn't taken from a patient. It was grown in culture. So really, I should see a nice, clean, white background, and I should only see these cells here. But I have some debris in the background. This is an old culture that has cells in it that have started to die and started to degenerate. And that's why I'm getting this strangely shaped, strangely colored material in that white background. It's also why I'm getting some cells staining even a dark purple and some cells staining a light pink. This is an old culture. I cannot determine the gram status of this. I can say it's a caucus, right? I could say it's a caucus, but I can't say what its gram status is. It's just not clear enough. This is a nice clear smear. These cells are all stained the same color. They are a bluish purple color. They are all stained the same color. And I have a nice clean background here of white. I don't have any debris on this smear. This one looks kind of bad. And again, this is what you'll see when you make a smear from cells in an old culture. So if I had to determine the gram status of these cells, I would take a few of them out of the culture tube with my inoculating loop using sterile technique, aseptic technique, and I would transfer them into a fresh broth tube. I would put them in the incubator and I would let them grow about 24 hours and I would try again. Take a look at this one. This one's even more obvious. This is a bacillus species. So this is one of our sporulators. This slide was made with an old culture. I know it's a little out of focus, by the way, so it's not your eyes, it's the slide. If you look around the slide, you can see cells that are not staining equally even within one cell. I can see within one cell, I can see purple and pink. And I can see some cells that are all pink. And I can see some cells that are all purple. And then I occasionally see a cell that has a white spot in the middle of it. You see that one? Here's another one. There's a big white area right in the middle of that cell. There's one right here that has a great big white spot, and up here. These cells are sporulating. The spores are where you see that white spot. The spores don't pick up the gram stain, so they stay white. But that's what's happening. That cell is going through the process, the natural process that we call sporulation. It's turning from a vegetative or a cellular life form into an inanimate spore. A spore, remember, is not alive in the traditional sense. It's got the DNA in there. It's got the important molecules in there that this cell has, but it's going to stop metabolizing. It's going to stop requiring any food. It's going to stop producing any waste material. It's just going to go into a state of suspended animation. And it will stay there until it finds itself in an environment that supports life, basically. A place that is warm and wet and has nutrients in it. There's only 2 genuses of bacteria that are capable of this. One is the Bacillus genus and one is the Clostridium genus. There are other bacteria that are not human pathogens that can also sporulate. Most of them live out in the soil. But in terms of human pathogens, there are only 2, the Bacillus genus and the Clostridium genus. This culture is old. It's too old. The cells are sporulating. I can't get a gram stain evaluation from this slide. I just can't. I can't say that it's gram positive or gram negative. I just can't tell. I would have to get a fresh culture and try again. Does everybody see that? Does everybody see the difference in those slides I showed you? When you get a good gram stain done, every cell on that slide is going to be approximately the same color. Now, the color will vary from organism to organism a little bit. Some organisms stain more blue, some organisms stain more purple, but across the smear, I'm going to see that they all picked up the same color. If I see that cells have not picked up all the same color, I know I have a problem. And I can't make a determination from that slide. I can't determine the gram status. I got a problem. Let's take a look at a clinical sample. So this was a throat swab. I should say this came from a throat swab. So the physician took a sterile swab, like the ones we've looked at, and put it in the back of this patient's pharynx and swirled it around a little bit, and then put it into a container with some transport media and sent it to the lab. And you, the technician in the lab, you took that swab and you spread it across a microscope slide to make a smear. And you let that smear dry, and then you heat fixed it, and then you gram stained it. So the background of this slide is not going to be that nice white color because you didn't grow this up in the lab in media. You literally took the swab as it arrived and you rolled it over the glass slide. And as a result, you got some material on here that came from the back of that patient's throat. You got some mucus on here and some other materials. That's why the background is sort of this pinkish color. And these big globs of pink over here, that's mucus. These darker things, though, these are important to us. So take a look. The physician wants to know two things. The physician wants to know, is there a microbe growing in the back of this patient's throat? And if it is, what's its gram status? And what would you tell that physician?
I would say it's gram positive and it's coccus
Very good.
and it's chain.
It is definitely chaining. Does everybody see the chains? Now, not every single cell is in a long chain. But look, I don't think I see one cell that's all by itself. So there's 2 cells chained there. There's 3 cells or 4 cells chained right there. They're chaining. And I have this nice long chain to confirm that. And I have another long chain right here and another long one right here. These cells are associating with each other. And you remember I said this is a classic feature of Streptococcus. Streptococcus likes to make these chains. So I would report that back to that physician and I would say, you have a gram-positive coccus on your swab and there's some chaining features. And the physician would say, it's probably strep. Now we would confirm that diagnosis, right, over the next day or two by culturing that organism and doing some tests to determine exactly what kind of strep it is. But you can report back to the physician that it looks like strep, and the physician knows, okay, good, I thought it was strep. I put the patient on an antibiotic, so I'm glad to hear it looks like a strep. Yeah, we often begin treatment for bacterial infections before we have all of our diagnostic information in. And that patient would then, keep taking the antibiotic because that's a strep. Very good. Good job. Does anybody have questions? Does anybody need to see anything again? Or remember, you have access to these. You'll have access to this recording, this meeting on our playlist when we're done today. So you can pull it back up and you can look at those again. You also have the slides, just the slides on Blackboard if you want to pull these pictures up again. Gram status is whether it's gram positive or gram negative and what the shape of the cell is. We can also get a little bit of information sometimes about arrangements of the cells. Not always, but sometimes. And sometimes that's a feature, that's a characteristic of an organism, and it gives us a little information about what it might be. We have to do a lot more testing, though, to be sure what we have. And that's what we'll be talking about through the semester, the kinds of tests that we do in order to confirm that we have that organism. Very good. Yeah, the only good thing about strep throat is that it's treatable, right? At least if you have strep throat, you know you can take an antibiotic and you'll feel better in a couple days. It's much worse when you have a viral throat infection and there's no treatment and you just have to suffer through it. All right, questions, comments? All right, don't forget, you have lab homework due by Sunday night. Don't forget you have your lab practical exam on Monday, and we have no meeting on Monday. No meeting on Monday. I won't see you again till Wednesday. All right. If you have any questions, you can message me. And I hope you all have a good weekend. I'll see you next time.
Thank you.
You're welcome.
Have a good weekend.
You too. Have a good day.
Thank you so much.
You're welcome.
Bye.
Bye-bye.